Hello everyone and welcome to Biology Forest. My name is Emma and I'm a biology teacher. Today I invite you to make some revision notes with me on the topic of enzymes. In this video I'll be making notes describing what an enzyme is, the lock and key model, as well as factors that affect the rate of enzyme action such as temperature and pH. At the end of the video there is a revision quiz covering the key points from these notes. Let's begin. What is an enzyme? An enzyme is a biological catalyst. A catalyst is something that speeds up a chemical reaction without being used up in the process. It is biological because it is found in living things. It is made of protein. What do enzymes do? Enzymes speed up chemical reactions in living organisms, allowing them to take place in the normal conditions inside living things. Enzymes catalyze thousands of different chemical reactions. These can be split into two main categories: building up large molecules from small ones, and breaking down large molecules into small ones. How do enzymes work? The way that enzymes work can be described as the lock and key model of enzyme action. In enzyme catalyzed reactions, the reactant is called a substrate. There are thousands of different enzymes, and each one has a specific 3D shape. The 3D shape is determined by the chemical bonds between the amino acids in the protein. Within the structure of the protein, there is a special area called the active site. The active site is the complementary or opposite shape to the substrate. Here I've drawn a diagram of an enzyme and a substrate. You can see the enzyme in green with its active site shown in pink. You can see that the active site is a very specific shape, and in this case it is the complementary shape to the substrate shown in blue. We can describe the active site as the lock, and the substrate, which can fit into that lock, is called the key. When the enzyme catalyzes the reaction, the active site and the substrate join together. When they do this, they form a structure called an enzyme-substrate complex. We can compare this to a lock on a door. Only a specific shaped key can fit in and unlock it. This is why there needs to be thousands of enzymes to catalyze thousands of reactions, because they all have different shaped active sites. Here I've drawn a diagram that represents how enzymes work. You can see on the left-hand side of the diagram we have the enzyme in green and the substrate in blue before the reaction begins. During the reaction, the enzyme and the substrate join together. We remember that this is called the enzyme-substrate complex. On the right-hand side of the diagram, the enzyme in green is unchanged at the end of the reaction. However, the substrate has been changed into the new products, which diffuse away from the enzyme. When the enzyme and substrate have bound together and formed the enzyme-substrate complex, that is when the catalysis or the reaction happens. This is when the product is made. After the reaction, the product breaks apart from the enzyme and diffuses away. The enzyme then becomes available to catalyze another reaction. The effect of temperature on enzyme action. Like all chemical reactions, enzyme catalyzed reactions are affected by temperature. When the temperature increases, the enzyme and substrate molecules gain kinetic energy. They move around faster, and this causes them to collide more. This means there are more enzyme-substrate complexes and more reactions. The temperature which the enzyme works fastest at is called the optimum temperature. This can be different for different types of enzymes. Enzymes in the human body, like amylase, have an optimum temperature of around 40 degrees Celsius. This is because your normal body temperature is 37 degrees Celsius. The optimum temperature is a bit higher, so the enzymes don't denature if you get a fever. If the temperature increases significantly, this puts strain on the bonds holding the amino acids in the enzyme into the correct 3D shape. Eventually, these bonds break and the enzyme changes shape. The enzyme is denatured. I've drawn a diagram here that represents how this works. You can see the enzyme on the right is a different shape to the enzyme on the left. The enzyme on the right is denatured. The substrate does not fit into the active site. Once the enzyme has denatured, the active site changes shape. This means that the substrate can no longer fit into the active site. When the temperature is higher than the optimum temperature for that enzyme, the rate of reaction slows down and eventually reaches zero at very high temperatures. The relationship between temperature and the rate of enzyme catalyzed reactions is shown on this graph. You can see in the first part of the graph, as the temperature increases, the rate of reaction also increases. We remember this is because the enzymes and the substrate gain kinetic energy and collide more, resulting in more reactions. The highest point of this graph is where the rate of reaction is fastest. That is called the optimum temperature. Above the optimum temperature, the rate of reaction decreases. That is because the enzymes are becoming denatured. At very high temperatures, all the enzymes are denatured and the rate of reaction is zero. The effect of pH on enzyme action. Just like with high temperatures, enzymes can become denatured at high and low pHs. Each enzyme has an optimum pH, where the rate of reaction is fastest. At pHs that are more acidic or alkaline than the optimum pH, the bonds holding together the amino acids in the enzyme into the correct shape break, and the enzyme becomes denatured. Enzymes in different parts of the body are adapted to have different optimum pHs. For example, pepsin, which is an enzyme found in stomach acid, has an optimum pH of 2. This graph shows the relationship between pH and rate of reaction for two different enzymes. Pepsin, which is the enzyme found in stomach acid, is shown here in green, and salivary amylase, which is an enzyme found in the mouth, is shown here in blue. You can see that they both have very different optimum pHs, Pepsin has an optimum pH where the rate of reaction is fastest at around 2, whereas salivary amylase has an optimum pH with the fastest rate of reaction at around pH 7. You can see for both enzymes that at pHs above and also below the optimum pH, that is when the rate of reaction goes down because the enzymes are becoming denatured. Eventually, the rate of reaction reaches 0. To end the video today, I've put together a short revision quiz covering some of the key points from this video. What you can do is you can pause the video and answer the questions, either in your notes or you can use flashcards. There are 11 questions in total. The answers can be found at the end of the video. Thank you for watching, everyone. I hope your revision went well today.